Today we're going to pick up with meiosis. We left off with mitosis in the last video. So mitosis can occur anywhere in the body. That's going to occur with all somatic cells. Remember, somatic cells are going to be any body cell. It could be a skin cell, a liver cell, a muscle cell. So any cell anywhere in the body. That's how those cells divide, through mitosis. Today we're going to talk about meiosis. This is only going to occur in one place. This occurs in either the sperm or in the egg. So this is going to happen with the gametes. So it starts with a diploid cell. A diploid cell is going to have 2 copies of each chromosome. 1 copy came from the mother, 1 copy came from the father. So in talking about humans, we had 23 sets of chromosomes. That's diploid sets. A total of 46, 23 from mom, 23 from dad. So with these, the specialized diploid cells in the gonads are going to be ovaries and testes. They're going to produce eggs and sperm. And so in the process, all these cells are going to go from diploid cells into haploid cells because you don't want to give 23 sets or 46 total chromosomes to the freshly fertilized egg and then have another 23 or 46 total coming from the father. That would give 92, and that would be way too many. So we're going to go over the process of meiosis, which is going to reduce those numbers of chromosomes, which are going to be found in the egg and in the sperm. So it's going to start, like we said, with a diploid cell. That means it's going to have 46 total chromosomes, or it might say 23 sets. So be careful, look at the wording. A homologous pair is what these are going to be called, or they could also be called homologues. So the maternal and paternal copies of a chromosome. So looking here, let's just say this could be chromosome 1. You have a maternal copy, which is the darker green, and you have the paternal copy, which is going to be the lighter green. And so this is going to be one from each parent. During replication, they are going to double up. So you end up with 2 of each, 2 from mom, 2 from dad. And at that point, they're referred to as sister chromatids. So each one of those chromosomes on the side is going to be referred to as a chromatid. And if they match up and they're identical, they're called sister chromatids. Or when you're talking about the set 1 from mom, 1 from dad, they could be referred to as homologues. So with meiosis, you need to reduce the number of chromosomes that are going to be found in the egg, and that is the goal of this process. It's going to be somewhat like mitosis, except for it's going to differ slightly because you're not just reducing the number in half to make a diploid cell, you're going to try to make a haploid cell from it. So it does have interphase. That's where each chromosome in a homologous pair is going to replicate to form a sister chromatid. Those chromatids are going to come together in the center. My tool. And where they come together in the center, that right there is going to be referred to as the centromere. The centromere is made up of smaller protein particles called kinetochores. And so they're together. First thing that happens during meiosis 1, we're going to take and we're going to split these 2 apart. So both of the sister chromatids from mom go to one side. Both of the sister chromatids from dad go to the other side. That's during the first cell division. Then, once you hit meiosis 2 and you go through the second cell division, each one of those sister chromatids is going to separate into their own cell. So you have 1 on each side there. So meiosis 1 creates 2 cells from 1 parent cell that you see here. After going through meiosis 2, you're going to have 4 daughter cells from the original 1 parent cell at the top. So just like with mitosis, we are going to go through the different steps that we went through with this phase, with meiosis. So interphase is going to be the preparation phase. So we double up the amount of genetic material. Remember, it is a diploid cell. When it doubles up, it's going to go from 46 chromosomes to a total of 92 chromosomes. Once it's ready, then those chromosomes are going to start to condense. So meiosis 1 division, we are separating first those homologues. Remember, mothers are going to go to one side, fathers are going to go to another. Prophase 1 is where the nuclear membrane starts to disappear, so you'll start to see that. You also have your centrioles starting to make their way to the poles. Also, one other important process is going to occur here. This, by far, is going to be the most complex of all of the processes and is a very important process. This is where crossing over occurs. Crossing over is when two of these homologues get close together and one leg of that chromosome on one side and one leg on the other side, so one from mom, one from dad, they get really close and they will actually swap genetic material. This is going to increase the chance of genetic variability in the offspring. And so crossing over is going to be very important, and you need to know that it happens in prophase 1. Metaphase 1 is going to be just like metaphase in mitosis. They line up across the center, except for in this case, you see how the spindle fiber comes out, connects in the center at that centromere, and is going to pull the entire set of sister chromatids to one side or to the other. So they line up across the middle. Think of middle and meta. So that's your metaphase. Next, you go into anaphase 1. That's going to be the beginning of the first cell division that occurs during meiosis. There, those homologules are going to be pulled apart towards the opposite side. So it's going to start to elongate that cell. You'll see the cytoplasm starting to go towards either one side or the other. And the maternal and paternal sister chromatids are going to be pulled to those ends in random assortment. So with the beginning of anaphase 1, you start to see where those homuncules are being pulled. So looking at the picture, notice how they're being pulled away from each other. Then it will actually start to pinch in on the sides with the cytoplasm. That's going to be where it comes in, is called a cleavage furrow. And eventually it will pinch in enough that it causes those cells to pop apart. That is referred to as cytokinesis when it actually pops apart. Also during this phase, you start to see that nuclear membrane reform. You see the chromosomes starting to loosen up some. They're not going to be as condensed, so it's going back into the form of chromatin. And so here's what telophase 1 looks like. Where this arrow is kind of pointing in here and here, those are referred to as the cleavage furrows. Notice how the spindle fibers are no longer coming out of these centrioles, and you start to see that nuclear membrane reforming. And so at the end of cytokinesis, that's what is referred to when you're doing cytoplasmic division. So that's when it divides into two cells. And then you will end up with two daughter cells. Then it's ready to go straight back into meiosis, and it's going to do a meiosis 2. So we go into what's known as prophase 2. There is no crossing over at this point. That's where, again, we're going to see that cell membrane start to break down. Nuclear membrane start to break down. You see these centrioles again going to each side or the pole. You start to see spindle fibers coming out. And the genetic material, once again, is going to coil tightly up. So it went from being chromatin to going back to being actual chromosomes again, where you can see them underneath the microscope. And so it doesn't stay in that interphase long. Next, they line up across the middle again. So you have meta-middle. This is going to be metaphase 2. Notice that As it lines up across the center, it's going to attach in to where that centromere was again, and it's going to break these sister chromatids apart. So one chromatid is going to go one way, and the other chromatid is going to go another way. And here you see that in anaphase 2. As the spindle starts to shorten, it's attached to that centromere, and it gets pulled to one side or to the other side. Next, we go into telophase 2. You start to see the nuclear membrane reform. Those chromosomes are going to start to loosen up and turn back into chromatin. And you see you have these cleavage furrows again, where it starts to pinch in. At the end of this, it's going to go through cytokinesis one more time, and you're going to end up with 4 daughter cells. So you started out with one parent cell with 92 chromosomes. You're going to end with 4 daughter cells having 23 chromosomes apiece. So 4 cells, 23 chromosomes each. Since they only have 23 chromosomes for a human, that's considered to be half of the genetic material needed, and so it's referred to as a haploid daughter cell or a haploid cell. Each one of these, because it was divided up, it could cross over, it's going to have a unique combination of DNA and traits. And so this is how we can mix up traits between 2 parents, and if they had lots of offspring, they still could all look unique and be individual. And so here's just a look of the process that we just talked about with meiosis and just a visual of what went on. So in summary, meiosis in animals occurs only in the gamete-producing cells. So this will happen at the ovaries or the testes when they're producing the gametes. It is preceded by DNA replication and consists of 2 rounds of cellular division, meiosis 1 and meiosis 2. One in which the homologous pairs of sister chromatids are going to separate, and then the second one, those sister chromatids will separate. And then the final product is going to be a diploid organism that has 4 haploid gametes. Now, male and female gametes are going to be produced in slightly different ways. Yes, they go through meiosis, but the end product is going to be slightly different. So the definition of male versus female is always going to be the same. The females produce the larger gamete, males produce the smaller, more motile gamete. The female gamete does get released from the ovaries, travels down the fallopian tubes or uterine tubes, and then it will reach the uterus where if it has been fertilized while in the tubes, it will implant. If it has not, it will then be shed with the monthly cycle. Now, that egg is going to have more cytoplasm and it's going to be much larger than the sperm. However, the sperm has a flagellum and it can move much further and go further distances on its own. So we went over gamete cell division. In females, you're going to have the diploid female cell. It's going to replicate its DNA. During meiosis 1, you have 2 homologues with a set of sister chromatids each. It's going to break off, and you have what's referred to as a polar body there. And so instead of going through meiosis with the set on the side here, that polar body is going to be reabsorbed by the body, and anything inside of it that the body can reuse, it will. Now you're going to take the second set, and it's going to go through a division, and it divides up into 2 daughter cells. And so this daughter cell here will end up becoming a polar body as well and be dissolved and reabsorbed by the body. This, however, will continue on, and this is the one that's going to make it to the actual egg. Now, the meiosis is going to be in an arrested phase once it starts to divide, and it's not going to go back into the actual division part until it's that time in the cycle. Then you'll have several eggs start to mature where they pick up and start to finish out their meiosis, and once one's matured, it gets released. The others are going to not complete their maturity and usually get reabsorbed by the body and the follicles. Every now and then, more than one can mature, and will be released around the same time. And if they both become fertilized, this is how you get twins that will not be identical. So in species with two sexes, including nearly every sexual reproducing plant and animal species, females are the sex that produces the larger of the gametes, and males produce the smaller of the gametes. Whether it's a male or female gamete that's being produced, each gamete ends up with just one copy of each chromosome. So crossing over and meiosis are going to be very important sources of genetic variation. Like we said, this is how you can end up with genetically varying individuals that come from the same parents. So crossing over, like we said, is when two of the homologues become close to each other. They're close enough that one leg will actually touch another leg, so it's kind of like crossing your arms or crossing your legs, and they switch genetic information. That is crossing over. Now, the area where they switched that genetic information is going to be referred to as the chiasma. And so that is that section that you see here, or the section where you saw them cross over. So that's your chiasma, and that's where the genetic information is going to be swapped. So although it doesn't create a new version of any traits, that crossing over during the first prophase of meiosis is going to create gametes with combinations of traits that may never existed before together. This variation is going to be very important for evolution. So what are the costs and benefits of sexual reproduction? Well, with sexual reproduction, you're going to have two individuals that come together that carry different genes, and the different genes they carry came from two parents before them that made that person or made that organism. When you put a male and female of each one of those together, you end up with varying genetic information. And so it's going to have alleles from two parents in each of your parents. They come together. Now you have this 4 different variations that you get to work with. On top of that, you're going to have the crossing over that occurs. And so then they can mix up their genes even more. And then you have reassortment. And so they randomly align up across the equator of that cell when it's going through division. And so it randomly pulls one to one side and one set of sister chromatids to the other side. And so sexual reproduction is going to lead to offsprings that are all genetically different from each other and from either parent in the 3 different ways that we just looked at. Bacteria are going to reproduce asexually, while most plants and animals, like we said, reproduce sexually. So which is going to be the better method? Well, it just actually depends on the environment and what's going on. If it is going to be an asexual reproduction, lots of times that can occur faster. Most bacteria will divide about every 20 minutes. Example, E. coli. And so you can go from 1 cell to 2 cells to millions of cells within a matter of days. Whereas with sexual reproduction, it does take much longer. Also, with asexual reproduction, it is going to be genetically identical to the parent. Sexual reproduction, it's going to be genetically different from the parent. So it depends on what environment it's in. If it's thriving in its environment, then that asexual reproduction would be more cost efficient. It doesn't take as much energy, and you're going to be able to create many organisms in a very short period of time. Sexual reproduction does have its disadvantages. Like with this that you see here, the black widow spider does often eat her mate and kill her mate after they mate together. And so she does attempt to kill him and eat him. Very rarely does he escape. So it is going to be dangerous for the male to mate and to carry on his genetic lineage. So it could cost them their life. Only half of the individual's alleles are going to be passed on. It's not going to pass all of its alleles on. It takes time and energy to find a partner. It can be risky to find that partner, and there's going to be lots of opportunities for mistakes. So, even though you get genetically unique individuals, sexual reproduction does have its disadvantages. Now, going into asexual reproduction, the advantages and disadvantages are going to be more or less reversed. It doesn't take a lot of time. They don't have to find a partner. They can reproduce very quickly, but they're going to be genetically identical. So if they come across an environmental problem, that organism may or may not survive it. If it comes across, let's say, a certain cleaner or something, that organism, if it's susceptible to that cleaner, like with this bacteria, then it might not survive. So how is sex going to be determined in humans? Well, sex is going to be determined by the 23rd set of chromosomes, and those are referred to as your sex chromosomes. Now, which parent determines the sex of their baby? Well, when you go in and you look, females or the egg can only have an X chromosome. Males, however, can have an X or they can have a Y sperm. So, who determines the sex of the baby? That will be the father, because he can give that X or he can give that Y chromosome to the egg. And when they come together, that will create either a male or a female. So in humans, like we said, there's 23 pairs of chromosomes in every somatic cell. Somatic cells, like we said, were regular body cells, and the gametes are going to be your sex cells. These can be divided into two different types, and there's going to be one pair of sex cells. So you can have an X or you can have a Y. 22 pairs of non-sex chromosomes have their own name. They're referred to as autosomes. So you have chromosomes 1 through 22 are autosomes, and the 23rd set is going to be the sex chromosomes. So there are going to be differences in these chromosomes. When looking at the sex chromosomes, notice that the X chromosome is going to be much larger than that Y chromosome. Now, with all genetic information, it is going to be stored on the chromosomes inside of the cell, but on chromosomes 1 through 22, it's going to be things such as, let's say, making skin or eyes or hair, things like that. It's not going to have to do with the specific sex of that organism. And so those enzymes and whatever it produces, it's not going to matter whether it's for a male or whether it's for a female, because those instructions are going to be the same for both sexes. So some genetic information, however, is going to instruct the body to develop into one sex or the other. And so that information that does that will be found in the sex chromosome. So it's going to be found in either the X or in the Y. So you inherit one copy from your mother and one copy from your father. And so the only one that you can get from mom is going to be that X chromosome. So if you're a male, you get the X from mom, that comes from the egg, and you're going to get a Y from the sperm, and that came from dad. If you're a female, you're going to get one X from mom, and you'll get one X from dad. So here's a visual looking at what the sex chromosomes look like in each sex. So with the females, you see the red chromosome there for their 23rd set, and it's 2 X's. With the male, you see the X and the Y, and so that's going to be its 23rd set. And so when that egg divides, it can only have an X chromosome in each egg. However, when the male gamete divides, it can either make half X and half Y. So you're going to end up with an X or a Y. When they come together, that's going to be how it's determined what sex a person is. So in humans, the sex chromosomes carry information that directs growing a fetus and developing it into a male or a female. Male if the Y chromosome is present, and female if the Y chromosome is not present. Now with these sex chromosomes, it is going to be important to know that it doesn't have essential things on there for life, such as, like we said, hair or eye production, things like that. These are going to be the ones that are solely there to help create things that are going to be sex determinant. Now we do have some sex-linked traits though that can be found on there, such as vision, color, and being able to see colors like that, and that will be a sex determinant trait. So sex determination depends on the sex of the chromosome inherited from the father. So the sex of the offspring is going to be determined in a variety of ways in non-human species. So if you look here at these birds, the mother determines the sex in a way that is similar to how fathers determine the sex in humans. So the mom could be either the W and the Z chromosome, so she can give either one to her offspring in the egg. However, the male is just going to be the Z and the Z, so he can only give the Z in his sperm. Ants, bees, and wasps, same way. Ants, bees, and wasps have their sex chromosomes determined by the chromosome set of individual possesses. So if it is double here, you see both of those, that's going to be a female. Just the one there, that's going to be a male. With turtles, it's going to be determinant upon the temperature. And so if the temperature is higher than the eggs that were laid during incubation, they're going to turn female. If it's a cooler temperature, then most of the eggs as they incubate will turn into male eggs. So a variety of methods are going to be used for sex determination across the world of plants and animals. They're going to include things like the presence or absence of sex chromosomes, the number of chromosome sets, and it could be the environmental factors such as incubation temperature. So, can the environment determine the sex of a turtle's offspring? Well, you can propose how to test the incubation temperature determines sex hypothesis. Well, you could put it into two different categories, and you could change the temperature. So when it comes to temperature, how does a lab differ from turtles' natural environments? Well, looking at this, the natural habitat experiment, half of the eggs are going to be incubated at a shaded site. The other half are going to be incubated at a sun-exposed site. The results of 100 eggs that are going to be hatched at the shaded site, all were going to be males. Of the 127 eggs that are going to be hatched in the sun-exposed site, 123 were going to be females. So the conclusions based on these results, well, if you put these together, it shows how much the climate really does influence what type of individuals you're going to end up with. So is temperature-dependent sex determination just a curiosity of nature, or are there any important implications for humans? Well, what would happen with these animals if you only had one sex that was being born without human intervention? How could we evaluate whether climate change might have significant biological consequences with these? So we know that things are getting warmer, and so with the warmer temperatures, we know that that would produce more female eggs. Unfortunately, you do need males to fertilize those eggs in order to have or to continue the species line. So we have to figure out what we're going to be able to do to change this and to help so these turtles do not go extinct. So observations of some lizard and turtle species reveal that sex determination is going to be influenced by the temperature during incubation of eggs. So both in the lab and in natural habitats, the cooler temperatures are going to be where more males are going to develop, and the warmer temperatures are going to be where you see more females develop. Climate change is predicted to bring an increase of about 4 degrees Celsius in North America in the next 100 years. So knowing that, we know that we're going to have a lot less males being produced if humans don't step in. So this increase is likely to have adverse effects on turtles with temperature-dependent sex determination and may signal for far greater impacts of climate change on biological systems. Deviations from the normal chromosome number are going to be leading to different types of problems that we see. Looking at this picture here, this is going to be called a karyotype. Karyotypes allow you to look at the different chromosomes, and not only can you compare them with size, but also you can look and see what genes are going to be found on each one of these. So starting up in the corner, you have chromosome 1. The next is going to be chromosomes 2. Then here you have chromosomes 3 and so forth. All of those are going to be referred to as autosomes. So All of these are going to be autosomes. The sex chromosomes are going to be found here in the corner. Now remember, the X chromosome is always going to be much larger than the Y chromosome. So you could look at this karyotyping here and know right away that this is going to be an X and a Y. So that's going to be the karyotype for a male. Down syndrome can be detected before birth. Karyotypes are going to reveal an individual's entire chromosome set. So with Down syndrome, it tends to increase at the age of about 35 in the mother. And the reason is because the genetic material in the egg at that point is 35 years old. However, with the sperm's going to be made fresh every so many weeks to about around every month if it's not used. Whatever's not used is going to be broken down and reabsorbed into the body, and a new fresh set of sperm will be made on demand. With the females though, we're born with all of the eggs we're ever going to have, and so our chromosomes that are going to be found inside of each one of those eggs is going to age with us. And so that's why the determinant of the mother's age, we start to see a lot of different genetic deformities go up to about 50% at around the age of 35, and they continue to increase as the mother gets older. So with a karyotype, that's a display of an individual's complete set of chromosomes, and that's what we looked at. And so if you look at this picture here, it shows you how they're divided by size. Usually it starts off with the largest ones as chromosome 1, and you continue on down. They start to get smaller and smaller, and then you reach your 23rd set, which are going to be your sex chromosomes. And those that you see there, it's an X and a Y again, and so that will be the karyotype of a male. One way in order to test the baby's genetic information is to use a process called amniocentesis. That's where they're going to use an ultrasound, and they watch as they insert a needle in. They insert that needle in and go through into the amniotic sac. And so that amniotic sac is going to contain a fluid that surrounds and protects the baby that the baby's floating in. Within the amniotic fluid, it's going to have the DNA or cells from that baby. So when they draw out the fluid, they usually need just about 2 tablespoons worth of fluid that they take out. They're going to then run it, analyze it, and look at the karyotype. Within that karyotype, they'll then be able to see if there's going to be any genetic deformities as far as chromosomal number. Next, you have what's referred to as chorionic villus sampling, or CVS. And this is going to be a process where they go in and they're going to remove some tissue from the placenta or from the chorion. Because the fetus and the placenta both develop from the same fertilized egg, their cells are going to contain the same genetic composition. So it can be done several weeks earlier into pregnancy, and usually we can get these results between the 10th and the 12th week. Now with Down syndrome, this is going to be one of the most common chromosomal disorders. It's named after John Langdon Down, the doctor who first described it in 1866. This is going to be where on the karyotype they're going to find an extra chromosome for chromosome 21, and that's one of the autosomes. It's going to happen to about 1 out of every 1,000 children that are born. Characteristics are going to include things like learning disabilities. It has a flat facial profile. Usually there's going to be heart defects. There's going to be increased susceptibility to respiratory problems. And this is simply looking at the karyotype for somebody with Down syndrome. And so we're looking at a boy again. We know that by looking at the sex chromosomes. We know this disorder because we see that there's going to be 3 chromosomes at set 21. The reason that the chromosomal genetic disorders occur is because of a process called non-disjunction. This is going to be the unequal distribution of chromosomes during meiosis. So what happens is you end up with an error during cell division as it creates those gametes, and either one cell's going to end up with 0 copies and the other cell's going to end up with 2 copies of a chromosome. And so when it goes and attaches in, you can have the spindle fiber attach into one, but it's also going to bring the other one with it. So you see both of those chromosomes going to the side. Then the cell's going to end up with more chromosomes. This one's going to end up with less. And so the daughter cells it produces has less, and these daughter cells that get produced will have more or have that extra chromosome in them. So in summary, the karyotype is a visual display of a complete set of chromosomes. A karyotype is going to be useful when you're diagnosing different chromosomal disorders. You take it basically from the fetal cells and you assess if there's going to be any type of abnormal chromosome structure there. The Down syndrome is going to be caused by having an extra copy of chromosome 21. So life is going to be possible with too many or too few chromosomes, but it's going to depend on which one that you have too many or too few of. So some common genetic disorders. You have what's referred to as Turner syndrome. That's where you have an X, but you do not have another X or a Y. And so with this one, they tend to be short in height. There tends to be webbed skin at the neck and shoulders. You have underdeveloped ovaries, and often these people are going to be sterile. And they do have some sort of learning difficulties or learning disabilities as they're going through school. Now, it could be because the egg had the chromosome, but the sperm did not have a sex chromosome, or the egg could have been missing the sex chromosome, and the sperm could have had one. The next one is going to be called Klinefelter syndrome. With Klinefelter syndrome, you end up with 2 X's and a Y. So because you have that Y, it will be genetically male, but that X is going to cause some difficulty in there. So it could be because the egg had 2 X's and you got the Y from the sperm, or the egg could be normal and you could have got an X and a Y from the sperm. Now with this syndrome, you're going to have underdeveloped testes, lower testosterone levels, usually they're going to be infertile, development of some female features. This means that sometimes the breast tissue does start to develop a little bit, so it looks like they have more of a chest. They can have rounded hips, they might have more of a rounded face, and just have some slightly female-looking features. They also tend to have long limbs, and they're slightly taller than average. With these people, lots of times they might not know that they have an extra chromosome, and they could be having problems reproducing or having a child with their partner, and it's not until they actually get tested that they find out they have Klinefelter syndrome. Then you have the XYY males, and the egg's always going to have the X, and it's going to be the sperm that was carrying 2 Ys. And so these are going to be taller than average males. They have moderate to severe acne, especially in teenage years. Intelligence sometimes going to be slightly lower than average, and sometimes are referred to as super males. Or this has also been called the Johnny Bravo syndrome. And so with this, these are going to be your people who tend to be really athletic. They tend to be really good at sports. They're usually very muscular and have a lot of testosterone. Then you could have the XXX females. The females here could be sterile, they may not, and so someone might not realize that they have this disorder, just like with the XYY and XXY, unless they get genetically tested. With this one for the females, there's not going to be any obvious physical or mental problems. Sometimes if they do find out they have the extra chromosome, they could be called the metafemales. So in summary, although it's usually fatal to have one too many or one too few non-sex chromosomes, individuals born with only a single chromosome, that is an X, or with an additional X or Y chromosome, usually survive, though often with physical and mental problems. That's going to end us for this unit. I will see you with Unit 7.